So welcome everybody. Welcome to California Master Beekeeper
Hello.
program.
Hi, Wendy.
Hello, good to see you, Gerhard.
Yes, indeed. Happy New Year, everybody.
Yes, Happy New Year to everybody. We are going to enjoy being educated this evening by Lauren Russert. She is in the Nino Bee
Bee Lab.
Lab. Yes, and she is working on researching the genetics of various lines of honeybees. So I
Very
am
cool.
going to turn it over to the expert, Lauren Russert. And as I mentioned before, we'd love to hear the story as to how you ended up doing what you're doing. So thanks for joining us this evening, Lauren.
Thanks for having me, everyone. As Wendy said, my name is Lauren Russert, and I am a first year PhD student. But I've been beekeeping for about at least 12 years now. I started, my interest flared with bees when I was in high school. A friend's mom asked if I wanted to go inside a hive and extract honey. So I gleefully joined them and I fell in love with bees, but I didn't know you could go and do it as a career. So everywhere I went, I would collect honey. I'd collect raspberry honey and blackberry and macadamia nut honey. Anytime I went somewhere, I would just get a different type of honey. Not really knowing that that wasn't necessarily the most important thing about bees. I just was so fascinated. And so I was a junior at Penn State, um, and I was going to animal science. So I wanted to be a vet my whole life, and that didn't work out. But I found out there was a honeybee class, and I excitedly signed up. And within a couple weeks, I was working for my professor in a pesticide research lab, looking at all the effects of pesticides on bees. And so I worked in her lab as a field technician for 3 years till 2011 at Penn State. And then in 2010, I took a little break from school and went to Brazil and worked with Africanized bees in Dave DeJong's lab in Sao Paulo, Brazil, and learned all about Africanized bees. And I just wanted to learn more and more. Went back to Penn State, finished up. I really stayed at Penn State so I could play an extra year of rugby, but you have to take a semester off to do it. um, and so I went back to Penn State, had one last semester, one last year of rugby, and one more field season with bees. And so I graduated Penn State and then went to Hawaii with the Hawaii Department of Agriculture and worked for the state apiary program, where I traveled the Hawaiian Islands, was essentially a beekeeper consultant, did national surveys, worked as a tech transfer team for BIP for the as a Hawaii tech transfer team. I did research projects, extracted honey, gave presentations, like you name it. I taught queen breeding classes. And then the last 2 years there, I was a state apiarist after my boss, Danielle Downey, had left to uh, be the director of PAM, Project Apis M. So I took over for her the last 2 years I was there. And I had always wanted to get a master's degree and further my education. So in 2017, I moved to North Carolina and got my master's on queen bee quality and honeybee genetics. And I had done the field work in Hawaii for that. So looking at how different queens on different islands, how their mating frequencies are. So how many drones they mate with on islands with rural mite and without, as well as looking at honeybee population genetics. Um, and I'll talk a little bit about that today, and I also did a stock acceptance project to look at how queen bees are accepted during the first 10 days of introduction. And so I I just love queens. I love bees, and I wanted to keep learning. I wanted to not only just master bees, but I wanted to be able to lead people. I wanted to be able to find a project and information that would help people in the long run. So now I'm in Alina's lab as a PhD student, and one of my big projects that I'm going to talk about today is stock performance. So how different honeybee stocks perform in a Northern California environment. And I haven't analyzed all my data, but I'll share with you what I have analyzed. And if anyone has any questions, long, short version of my bee experience, but I'm excited to be here today with everyone. And yeah, please let me know if there's any questions throughout or afterward. Okay, so I'm going to share my screen. First, does anyone have questions before I move on? No? Okay. Okay, can everyone see this? Yes? Okay, great. Okay, hi everyone. Today's January 11th, Monday, and I'll be talking about evaluating honeybee stock performance. So for the problem and significance, I'm going to see if I can move this. There we go. Okay. For the problem and significance, there's high annual colony losses yearly. Many of us know this already. Beekeepers continuously replace colonies every year, as well as queens. These high losses often are due to colony or queen performance. Queens are often underperforming and need to be replaced below her maximum reproductive lifespan. And queen quality is an essential driver for colony health and productivity. In addition, over 1 million queens are produced each year, of which 900,000 roughly come from 500 breeder queens. Which is a very small pool of queens that these come from. So looking at these different stocks is important, and especially because we don't have a lot of genetics in the US. Beekeepers largely base what's acceptable for a colony based on previous experience with that stock or word of mouth through a breeding program. And there's just not a lot of information out there. Queen and colony performance have a lot of potential to induce long-lasting colony level changes in their performance. And this, as I said, there's not a lot out there, and this is an important study to just share with beekeepers what would be relevant to them for whatever kind of queen or honeybee hive they have. And part of what I'm doing is assessing different stocks to look at the efficiency and management to better understand how to improve beekeeper management and practices across the different stocks and in different environments, to characterize and compare multiple lines of honeybee stocks, especially newly established lines, and to evaluate colonies for their spring growth, queen rearing abilities, and subsequent queen quality, honey production, monitoring for varroa, viral loads, as well as disease and pesticide resistance. So all of these things are really important when it comes to management. So I have different objectives for the first year and second year. For this first year, it's just evaluating the 6 US queen lines that these queens are. And just a reminder that honeybees are not native to North America. So even though I say these are US queen lines, that I'm saying that these are queen lines that are in the US. A lot of these lines are from different parts of the world, but we've had them here and bred them within the US. So for this first year, I assessed the 6 different lines, recorded their colony performance, including brood pattern, production, honey, nectar, and pollen production, hygienic production, behavior, how many queen cells they're producing, and then the presence of disease. And then for the second year, I'll be doing the same exact thing, But in addition, looking at pest and disease tolerance and resistance, including bacterial diseases and or pesticide in both lab and field. So there's many different diseases for virus. There's many different diseases such as American and European foul brood, and seeing which lines might be resistant to those, as well as viral loads. Are there any lines that are more susceptible or resistant to deformed wing virus or black queen cell virus, nosema, just different viruses, diseases, and fungal pathogens that might occur. And then another thing is assessing the daughters of these queens as well as the quality of the drone. So we would be sending these off to North Carolina to look at sperm viability, sperm count, the quality of them. And then fourth objective for the project as a whole, but that would hopefully be done next year, is looking at the interactions of acceptance among the different lines. So I was at North Carolina, let's say a Russian queen in an Italian hive, how well is that Russian queen accepted? So that would be another thing to look at the supersedure rates inside the colony. And that fourth objective would be if I can get everything else done. So these are the 6 honeybee lines that I'm working with. I'm going to give you a brief recap of what all these mean, what all these different stocks represent or are known for. So Italians, the queens are from Northern California. They're commonly used in the US. They're one of the most common queens found in beekeepers' apiaries. They're known to be very gentle and good honey and brood producers. The Carniolan queens are from Hawaii. They're very adaptable to different conditions, overwinter well. They're highly productive when there are good conditions. Caucasians, we received those from a breeder in Northern California. Those are really good propolis collectors, very docile, strong brood pattern, possibly pesticide tolerant. And then the VSH and pole lines, so those are 2 different ones that we're looking at, are from the Gila, Hawaii and Baton Rouge, Louisiana, and they're known for their hygienic behavior, bred for mite resistance, and generally docile. And then Saskatraz, we received those from a breeder in California, but they're originally from Saskatchewan, Canada. They're bred for honey production, overwintering ability, temperament, tracheomite resistant, varroa tolerance and resistance, and low brood diseases. So these are the 6 different types of stocks. And as I said, these are a lot of these traits that I described, a lot of them are anecdotal. So one of the biggest purposes of this study is to quantify all of this. Like, are Italians actually really good honey producers and brood? And are Carniolans adaptable? And so just seeing, and as I mentioned earlier, these are just based on Northern California conditions. So a beekeeper in Maryland might get an Italian and it might act differently. So this is all very regional specific. And also the breeder, just because I have an Italian from one breeder doesn't mean that all Italians from other breeders are like this. So these are all things that I'm going to consider and look at when I analyze the data and write up the reports on this. But this is just giving a general overview of quantifying these different lines. So for this project, I managed 76 different colonies. I started with packages in mid-April, and then in early May, I introduced queens. For each line, I had 12 or 13 different colonies, and I ended up checking the bees every 2 to 3 weeks, and then doing an assessment once per month. And I'm gonna describe how I did these assessments. And the colonies were fed sugar syrup as needed. So as I said, there were 4 assessments total in June, July, August, and September. Each assessment, for each assessment, I looked at the frame of bees before smoking the hive. And then on a scale of 1 to 10, I looked at the brood, honey, nectar, and the empty frames. I also collected nurse and forager bees that I'll hopefully be looking at either virus levels or gene expression to see how the bees differ. And I collected a mite sample in ethanol using a third cup scoop, so roughly 300 bees. And I looked at the percentages that we'll talk about later. In addition, I also, on top of all that, I looked at queen cell production, disease, queen death, and when they might have died. And then on a scale of 1 to 5, I looked at the color, pattern, and temperament. And on the right here, this is on the top, this brood pattern, that might be, I'd consider that close to a 4.5. So just to give you an idea of what I might be doing. So the top right picture, I'd give it a pattern of 4.5. And I apologize, I meant to add pictures of how I scale the different things. And then the color of this, you can see some really light bees, and then you see some darker bees. So this, the lighter the bee would be closer to a 1 or 2, where the darker the bee would be closer to a 4 or 5. So I'd rate these somewhere in the middle, close to a 3. Italians often are closer to a 2, whereas Carniolans are close to a 3. And the Caucasians and Saskatraz are closer to 4. And this is when I would do assessments with BIP. These aren't necessarily BIP assessments because I'm the one assessing them right now, but we would do similar assessments with BIP with the beekeepers that we would inspect. And so a lot of this is based on that, but using my own criteria. And then the bottom right picture is a picture of hygienic testing, where I would count the number of cells within a 3-inch ring of PVC for brood. And then you remove, once you pour the liquid nitrogen in, you go back 24, 48, and 72 hours later, and you see how many of those cells the bees removed. So seeing how hygienic these bees are is really important too. So if you have a colony that's high, that has really high mite loads, how well are those bees able to handle it? So that's a really important measure that that I'm looking at as well. So when we look at the results of these, and don't get too confused, I know sometimes graphs are really confusing to look at, so I'm going to break these down. On the left here, we have for each assessment, how the different colonies performed, the different lines. And then on the right, we have the total average frames of bees for each stock overall. And on the left here, there was no significant difference for individual assessments. So that means that all of them were using an ANOVA. They all rose and fell during different times. And I'm going to go through that step by step in the next slide. And then I'll explain the other graph next. So for this graph, the stock with the least number of frames of bees. So frames of bees is when you're looking at when you're looking at a hive, you open it up and say you have a 10 frame hive and there's 5 frames that have bees on each side. That would be 5 frames of bees. So this is, it essentially measures the colony growth over time. And so this graph on the left shows during each assessment, how did each of these different lines, the average of them, how did they perform during that assessment? And so I'm just going to walk through this. So the stock with the least number of frames of bees throughout the 4 assessment were the pole, which is this yellow line at the very bottom. So the pole line had the least number of frames of bees, followed by the Saskatraz. And so these colonies stayed a little smaller than the rest. And then the next point with this graph is that this green line are the Caucasian, and they were in the lead for having the highest amount of frames of bees for the first 3 assessments. So assessment 1, assessment 2, and assessment 3, they were above all the rest. But then by assessment 4, they had dropped to 4th place, to 4th in line, where their frames of bees went down below the other 3. And then for the Carniolan, Caucasian, and the Hilo VSH, so these 3 in the middle, those were all those were all statistically very similar. And so I think the main takeaway from this graph is that the Poland Saskatraz had the lowest number, those colonies stayed the smallest. The Caucasian started off really strong and was the highest until the end, and then they just went down. And then the Carniolan, Caucasian, and Hilo VSH were in the middle, and they stayed consistent and with each other throughout the whole time. And then if we look at the overall frames of bees, so these numbers at the top are what the average frames of bees, if you took all the assessments for all those lines, this is what this is what I saw overall. So the Caucasian, as I said, they had the highest, and even though they dropped during the 4th assessment, still had the highest frames of bees, followed by Hilo, Sage, Carniolan, and Italian. And then the Saskatraz and Pole, which we had mentioned earlier, those were the ones with the lowest frames of bees. So um, this the largest colony, and then it goes all the way down to the smallest colonies. And then we have the mite levels. And again, I'm going to try to explain this in a way that makes sense. For all the assessments, there were significant differences for the 2nd and the 3rd assessment. So this is assessment 2 over here. Oh, this is assessment 2 and then assessment 3. So these had the biggest differences between the different lines, and then not as big a difference for assessment 1 and assessment 4. And then I'm going to walk through this varroa mite loads graph on its own also. And there were significant differences for these, all the stocks overall. So when we look at the mite levels during the different assessments, all stocks started out with mite levels at or below 2%. So even though these are a lot of numbers, if you look at the numbers on the y-axis right here, these are the percentage of mites. So all started out with less than 2%. Even though some of them look a lot higher than others. The pole and the VSH, so this yellow line and this pink line, those only both slightly increased. So throughout the entire summer until early September, actually was it mid to late September, these all stayed below 3%. And a note with all of these that none of these hives were treated. And so without ever being treated, the pole and BSH, which are the hygienic bees, those ones stayed pretty low. The Carniolan stock, which is this red line, they increased to 6.1%, starting at around 1.1%. They went all the way up to 6.18. And then the remaining stocks averaged mite levels at over 12%, which is pretty high. The mite level averages for all stocks remain below treatment threshold until the 3rd or 4th assessment. So all of them stayed at or below 3% until later on in the season. So this is important to know, especially management wise. I purposely did not treat these so that I could see if there were some stocks that were more resistant to mite levels than others. But if these had been my personal hives, when you see certain thresholds, you want to make sure that you, when you're monitoring, if they go above the threshold of 2 or 3%, whatever you're setting for yourself for a healthy level, you want to make sure you get to those bees before you treat the bees before they skyrocket. And then the average mite levels for the stocks were also increased. They increased slightly just by 1 or 2 hives. So even though these might be at 13 or 14%, it might have been 2 of the 12 hives that made them go up. So I'm still looking at all the data and seeing how all of this fleshes out. So um, any data that I give you guys, this is my personal raw data. So if you could not go spreading it to everyone, especially since it's all preliminary. I don't want to give a bad name to a certain line over another line. So this is all just general information right now. But the main takeaway from this is that the supposedly hygienic queens, the VSH and Paul lines, those stayed relatively low. Carniolan was in the middle, and then the Saskatraz, Italian, and Caucasian, both, all 3 of them increased pretty rapidly towards the end. And then when we look at the average varroa levels for all the different hives for each stock, the Caucasian had the highest, Saskatraz next, Italian, and then Carniolan, Pole, and VSH. So again, as I said, the Pole and VSH had the lowest. The middle one was the Carniolan, Italian, Saskatraz, and Caucasian were the next highest. And as I said, please don't go spreading this to other people that Caucasian have the highest mite levels, because that's not necessarily accurate. This just needs to be fleshed out more. And then when we look at brood levels, so for the rest of the results, none of them are statistically significant. So I'm just going to go over them briefly. So for brood, there was no statistic difference. Caucasian did have the highest, whereas pole line had the lowest. So if we go back to this last graph, even though Caucasian had the highest mite levels, and pole line had the lowest mite levels, or almost in the lower mite levels, you can see that the brood here for the Caucasians was the highest. So that could also relate to the number of mites. So more bees could mean more mites. And the same, you don't have as many mites, so you might not, that might be due to not having as much brood. So that's something to keep in mind. And then the other 4 lines were in the middle. And then for honey and nectar, so we have honey on the left and nectar on the right. For the average honey hives, both the Caucasian and Carniolan hives, those had higher honey production levels, at least based on the number of frames. I don't have a weight for these hives, but the amount of honey that was in the hive. And then if you look at the Italians and Saskatraz for these assessments, they had the lowest overall. And then for the nectar, the Carniolans had the highest amount of nectar, whereas the VSH had the lowest. And again, as I mentioned, there's no statistically significant difference between the different stocks for these. And then for the amount of pollen, Carniolan, there's still no statistically significant difference, but Carniolan did collect more than the other lines. And then if you look at Saskatraz, Saskatraz collected the least, and then all the other ones are in the middle. So this just means that the average of the carniolan is right here. So it's only slightly above all the other ones. That's why it's not statistically significant. And then there were a couple that might have been higher right here than the rest. And you guys have probably seen this in your other hives too. You could have 10 different hives, and one of them, one or two of them, even though they're from the same queen mother, they might be super producers of something. So even though the average of your whole apiary is a certain amount, you might get a couple that are much higher than the rest. And that's something that I'm going to flesh out more in the data. So, yeah.
Are all these hives kept in the same location?
They are. They're all kept right near our bee biology, about a mile from our bee biology lab. And inside the apiary, they're randomly placed. And so they're almost in a couple U-shaped half circles inside the same apiary.
Okay, so they all have access to the same resources.
Correct. Yeah. Yeah, and one of the last slides I'm going to talk about are things that might have happened in the beginning or throughout the project that led to either smaller colonies or why some might be larger than others. And it's also why we're going to be doing 2 or 3 years of this to get more data to look to see how they perform over different amounts of time. Okay, so for queen losses, there were queen losses with all stocks. There was at least one queen loss per stock. For Carniolan and Caucasian, there was one queen loss during the 4th assessment. For the pole stock, there was one queen loss by assessment 1. Saskatraz had one queen loss during assessment 1. Hilo had 2 queen losses, one during assessment 1 and one during assessment 3. An Italian had 1 queen loss during assessment 2 and 3 queen losses during assessment 4. And all of these different stocks, there were queen losses for multiple reasons. Some rejected the queen within a couple weeks. So like the Saskatraz, there was a queen that was rejected and the hive refused to requeen. They wouldn't do anything to accept a new queen. There were some that from the beginning, there was another type of stock that had continuous queen cells. That they just wanted to requeen that queen. So it could have, and then some might have been my fault. If I accidentally squished a queen, I'm going to own up to that because it happens. But overall, queen loss was pretty minimal for the stocks. And then when it comes to colony losses, for throughout the hive, there were 5 losses up until the 4th assessment. For carnies by assessment 3, so it was money, and it eventually was lost. The Saskatraz had 1 loss by assessment 3. The Hilo VSH, no direct colony losses. So once a queen for those was lost, we didn't have any more, and so I had to convert those into pole line colonies, which weren't used in the data. And then for the Italians, there were 3 losses during the experiment by assessment 4. And so they all had different reasons for losses. Like I said, the Saskatraz, the colony just didn't want to requeen itself. The pole line, it was just a small colony. The Italian, they ended up getting, a couple of them ended up having higher mite levels. And so there were just different reasons for colony losses. as I said, potential causes, lack of queen acceptance, ongoing fires, and the increased number of colonies introduced during that area. I don't know if any of you guys experience fires around your hives, but I think it directly, I don't have scientific evidence, but I do think that they had a large part in whether the colonies grew or shrunk, or whether how the queen was performing. That's a whole nother conversation, but there were lots of fires around the area. Lack of foraging resources. This year it was, and I'm more newer to California beekeeping, but there just weren't a lot of resources. So having to feed the colonies more. Pesticide exposure. There, I know that my apiary is right next to a vineyard. And so not knowing whether the bees were exposed to anything there or on any other agriculture area. In the very beginning of establishing these hives, I had the top hive can feeders. And when I put those on the hives, sometimes I'd come back and the bees were just drenched, and I might see a few hundred bees dead. And it's heartbreaking. And it's a lesson for me for the next year for the type of feeder to use. Colony management might be a cause for loss, for colony or queen loss. Scarcity of queens a bit available. And so these are just potential losses that might have happened. So some of the improvements to this, so for these colonies, I didn't treat the packages in the beginning, but retrospectively, that might have given them a better chance in the beginning to just knock down mite levels. As I said, having a different type of feeder for syrup instead of the top feeders. Scheduling more orders of queens throughout, having at least one backup hive for each type of queen, and allowing colonies to build up more in the spring before starting assessments, which is tricky with packages and the field season. So with research, it's really hard to juggle it all and still come out with a healthy hive, because we're constantly sampling these hives and testing them for what's different things. So trying to find a way to incorporate all of it to work for looking at it from the management side and the research side is really important. So what's next with this project? Continuing to manage the hives. Currently, there are about 20 of these hives left, which is, so after the fourth assessment, I've been going back and looking at the hives and looking at the colony losses, and we've lost about 75% of them, which is a really high number, but it's also good to see what happens when you leave the hives to their own devices and you don't manage them, and you well, I was managing them, but when you don't treat them for anything. And then I still need to analyze the rest of the data. So I gave you guys a little piece of the data, but I still need to finish analyzing the hygienic testing, looking at diseases, analyzing queen cell numbers, and just finishing analyzing all the data. So there's a lot of work that still needs to get done. And a summary of everything, for the frames of bees, there was a statistically significant difference for overall frames of bees. There was not a statistically significant difference for frames of bees for each assessment. For the mites, there was a statistically significant difference for mite levels overall. There was a significantly significant difference for mite levels for assessments 2 and 3, but not the others. And there was no statistically significant difference for overall brood honey, nectar, or pollen amounts. And when we look at the summary for the queen and colony losses, for the queen losses, there were 8 total queens lost, at least one from each line. And then the Hilo queens had 2 lost and the Italian had 4. And then for the colony losses, there were 5 total. Carniolan, Caucasian, and the Hilo had 0, and the Paul Saskatraz had 1, and then Italian had 3 losses. And this is prior, during the assessments, not afterward. And with all of this information, how can this help master beekeepers? So why is this even relevant to you guys? Well, evaluating which stocks are better for you and your beekeeping styles. So whether you're a backyard beekeeper or a commercial beekeeper, do you want to enter the hive? Do you collect honey just for that season? As we stock, you might get a really high brood or honey amount, but then the colony dies in the end. So, or how do you want to manage all of it? Looking at different temperament brood honey production for the hives, if you want a certain type of bee, or if you live in Southern California versus Northern California. For a future project, looking at, are certain queens easier to be accepted? If you have an Africanized hive, which queen do you get to be accepted more? Which hives need less mite treatment overall? And then in addition, looking at management. Management is so big when it comes to beekeeping. Most people that don't know bees, they think they don't realize how much effort goes into beekeeping. So looking at monitoring for varroa, when are the critical time points for these different stocks? So there's an overall best management practices, but what if we could make that more specific? It's if someone had varroa level Italian bees or carniolan bees, say it might be better to treat carniolan bees at a certain time of year versus other stocks. Learning the best routine colony inspections, best ways to feed the bees, and a lot of general management, how to overwinter bees wisely. And a lesson from all of this is, just because a colony is strong early on, you might have a colony strong early on, but that doesn't mean it's going to remain strong or be the strongest or even survive later on. So not getting our hopes up early on in the season that it's going to be a really great year. And it might be a really great year, but how do you mitigate all of that and allow for it to be a great year? And if you want that to remain a strong hive, what are the steps to do that? And seeing why colonies may not make it, or the reality of queen acceptance. So why did the colony not accept the queen after trying 3 times? So just looking at different aspects of that. And in addition to this project, I'm hopefully going to be working on queen introduction and acceptance among different stocks, which would also benefit California for looking at different ways to introduce European queens into Africanized hives. And just in general, how do you get higher queen acceptance rates? And then I'm also currently working with a project with the Buck Institute, looking at queen and worker bee aging, and the different stages of the development during different time points, and how the genetics and transcriptomes of these bees look over time. So with that being said, I want to thank everyone that helped with this project, and everyone in the lab, the USDA, the California State Beekeepers Association, all the queen breeders, and just everyone that helped me along the way. And if there are any questions, I am happy to take them.
Thank you. Thank you so much. That was very enlightening. And I appreciate your caveat around its raw data and it's not something that we can go out and start professing as truth, science-based
Mhm.
truth, because you're still in the process of actually doing the research, right, and collecting more data. And my question is, and I likely wasn't paying enough attention at the very beginning. I was curious as to how many hives you had of each of the lines. I visualized the whole U in the apiary, right?
Yeah.
So, how many hives of each?
So I have 12 or 13 of each. So
Wow.
I had 12 of everything at least. And then if I was able to get an extra colony, so for the Hilo VSH, we were only sent 12.
Mhm.
And so we weren't able to get that 13th hive. So for some of them, we had 12 and some of them 13
Wow.
hives.
Wow. Okay.
Yeah. And 76 hives total.
Yeah, and it was interesting to me that the Italians ended up losing the most queens and the most colonies, and I
Oh,
guess that's relative, right?
right.
Yeah. So,
Yeah, and it's all fascinating. And I think something that wasn't so surprising were the mite levels for the VSH and Hilo, or
mhm,
the VSH Hilo and the pole, which is really great. It's confirming. Um, but looking at all the other lines, and especially like Italians are queens that a lot of people use. So, is that really the best line for someone in all these different environments, or would there be a better line that's suited for them? I know a lot of commercial queen breeders use Italian, and that's great. But what about the backyard beekeeper? And what might be more like are carniolans better? Like, are they actually more adaptable? So can they survive a dearth better? Um, and so what's best for each beekeeper?
yeah, yeah, that's really important. And to my knowledge, there's not really anything out there
Mm-mm.
on that, is there?
No, there might be one or two papers, and it's more about a specific stock. So I've read a paper on Italians, but there's not a lot of other research on there on the different types of queens and bees.
Mm, wow. Well, good
Mhm.
work. Really great work. Thank you for sharing it. And I'm wondering if there are any other questions for Lauren this evening.
Any
If not,
questions?
sure.
No, I said I don't. I've got bees out on an island here local, 15 years, and the bees have done really good. And this last year, a commercial beekeeper was allowed to come in and dump like 800 hives out there.
Wow.
Bees have never been treated, never been messed with. I've got 2 of them in boxes, the swarms. The rest of them keep going into the wood duck boxes and end up dying.
Hmm.
And so this year I'm going out with some swarm traps to put in the locations where they've gone to. But I do have 2 hives out there right now. They have not been treated. They're doing good. I will find out more when I go out this week to look at them. But I haven't had any problems with those, and I'm waiting to see what's going to happen, being they brought in the commercial bees. There's a guy there was saying, "Oh, well, we can treat your bees too," and it's like these bees haven't been touched.
Right. And that's a really, your name's Ginger, right?
Yes.
Yeah, that's a really important point, Ginger, because there's a lot of areas. So, a good example is Africa, where there's bees and there's mites, and they coexist. And so they've never treated their hives there, and everyone's on the same mentality with it, where they don't treat their hives, but the mites also don't become resistant to all these different chemicals that are being thrown at them. So their super mites aren't being bred out there. So it very well, the island where your bees are at could be something similar, where they've created a relationship where the bees are able to survive with high mite levels, or with mites in general, whether they have higher mite levels or not, or virus levels. The mites there might not be hurting the bees there as much. So it'll be really interesting. I'm curious what you have to say after you check the bees with the commercial queen breeder, because that changes the whole, what's the proper word? Not ecosystem, but it just changes the balance of everything with someone else moving in there.
Yes, and I know, and I said the what I'm gonna call the mother hive has been in the same place for over 15 years. You can't physically get to it.
That's incredible.
It sits behind a 400 amp disconnect on a wall. On the inside, you can walk up and you can watch the bees coming and going. On the outside, you're 40 feet up and there's a hillside behind you.
Wow.
House
Do you
for the
know,
island.
do you know if you had mites there before the commercial beekeeper came in?
I haven't treated for mites out there. I've kept an eye on that hive. That's the hive I get the swarms out of.
Right.
As far as I know, there haven't, I mean, for 15 years, they've been in the same location. That one
Right.
hive has been.
Because if no one's ever monitored for it, that might have been an area with no varroa either. And so that's the picture that I have on the screen. This is on the island of Molokai. And that hive right there, like there's no mites on that island. So that could have been the situation also. So that'd be interesting to see if your colonies make it and yeah, how it changes everything.
Yeah, that's what I'm waiting to take a look. I'm gonna go out there and take a look at it. I know when I drove by the other side of the island, because I took a trip all the way around it to take a look at his hives out there, he had put them on the ground.
Mm-hmm.
I saw a lot of his hives were not doing very well with the ants.
Mm-hmm.
I don't know if he realized that there were wild pigs out there as well. So I haven't been all the way around again, but if any of his hives smelled good, I'm sure the pigs would have gotten into those because they have now mowed all the corn down. Because it's basically the only thing that's out there is corn, the blackberries. There are some, I don't think you call it wild radish or wild mustard,
Yeah.
Mm-hmm.
the little purple and white flowers, and then water hyacinth. And the honey that comes out of there is really light. It's got a different flavor to it.
That's
Wow.
amazing.
Yeah. Yeah. Thanks for that. Thanks for that, Ginger. Great story. And we'll be interested to hear the report
Mm-hmm.
about that hive. So next time we're together and you've visited, please let us know.
And now the sun's shining, I'm going to go out there.
Yeah. Yeah. So that brings us to the top of the hour. And we thank you, Lauren. We thank you. We'll be putting this in behind the portal, the members portal. So it won't be shared publicly. It'll be shared with the members group with all the caveats. And it is also a great webinar from the perspective for any master beekeepers who are interested in doing a research project, basically, because you took us through your way of thinking, and you took us through everything that you were able to analyze, the four various things that you wanted to you put the hypotheses out there and the answers that you wanted to all your questions. So it's also a great roadmap for that.
Yeah.
And we look forward to hearing back from you when all of the data is in. That'll
Yeah.
be fun. And when this paper's published. So
Yeah, thank you. And if anyone, just a little plug, my first publication as a first author is coming out on the 14th about for my master's. So keep an eye out on that for
on
for queen mating in Hawaii.
Thursday,
Yep, Thursday.
we'll share it and send it
Oh.
to me and I will post it. Definitely. Thank you.
Yeah.
Yeah. Congratulations.
Yeah, thank you. And thank you
Yeah.
guys for having me. This is exciting. And
Yeah.
I'm excited to share more on my project and what I discover and how I can help you guys.
Yeah, we appreciate you. Thank you so much for your time this evening. Really appreciate it,
thank you.
especially, knowing that you've got the family in the background, etc. So thank
Mhm.
you for taking the time to be with us this evening. And
Thank you.
yeah, we'll catch
I
you at the lab.
apologize if there were any noises.
No.
I can hear my husband playing guitar downstairs.
Yeah, and the baby's laughing and giggling. There's
Yeah,
nothing
crap.
sweeter. So there you go.
Yeah.
Thanks
Well, thank
again.
you guys.
Have a great evening.
Yeah, thank you.
Be well.
Thank
Bye.
you.
Bye.
Bye.